You didn't do that section, did you? Oh, okay.
That is actually an attached or detached earlobe. Okay, everyone has an earlobe. It's just some people genetically, it is a gene that gets passed down that will cause someone's earlobe to be attached. Other than detached. Okay? Just to clear that up about the earlobe. Everyone has one.
Thank goodness, but no, it doesn't look like that one.
Okay, so we're gonna go back to inside of the inner ear. We're gonna make our way into the inner ear. So we started out here at the auricle, the ear, then inside the external acoustic meatus, and then we were at the tympanic membrane. Behind the tympanic membrane is the middle ear. So all of this is the external ear. Again, this is deep in the petrous portion of the temporal bone. So now we're behind the tympanic membrane. This is the eardrum. Tympanic membrane anatomically. Behind that, we have the three smallest bones in the body. We're in the middle ear. Malleus, incus, stapes. Stapes is right up on what's called the oval or window. Below it and more anterior to it is the round window. How we leave the inner ear. The oval window is how we get into the inner ear. Okay? All right. Stapes is that small bone that's right up on the oval window. Then we get into the inner ear where we find the bony labyrinth. The bony labyrinth. Part of the bony labyrinth is called the vestibule. That has semicircular canals. The other, and this is devoted to equilibrium. The other part of the bony labyrinth is the cochlea. Looks like a seashell. This is devoted to hearing. So we're at the bony labyrinth of the inner ear. Inside of the bony labyrinth is the membranous labyrinth. The bony labyrinth is the vestibule with those semicircular canals and the cochlea. It is filled with perilymph, the bony labyrinth, okay, which is similar to the cerebrospinal fluid. And then we go into the membranous labyrinth that's surrounded by this perilymph, but follows the contours of the bony labyrinth. So it coils within the bony labyrinth, surrounded by the perilymph inside of the bony labyrinth. But it is filled, the membranous labyrinth is filled with another fluid, endolymph. And that's similar to cytoplasm in our cells. The makeup of that. The membranous labyrinth again travels the contours of the inner contours of the bony labyrinth. And it consists of the sacs and ducts. Here are the bony labyrinth that sits inside the petrous portion of the temporal bone. On the outside is the bony labyrinth, where we have the vestibule and the semicircular canals devoted to equilibrium, and then the cochlea devoted to hearing. Inside of the bony labyrinth is a fluid called perilymph. Surrounding the perilymph is kind of surrounding the inner membranous labyrinth that follows the contours of the bony labyrinth. Like like so. Here, the bony labyrinth is removed, and you can see the membranous labyrinth that takes the same shape as the bony labyrinth because it's traveling through the bony labyrinth. The membranous labyrinth is filled with a different fluid, endolymph. Okay. So here we have a model of the inner ear. We have the vestibular organ. Okay, this structure in here, and the semicircular canals devoted to equilibrium. Then we have the cochlea, it's like a seashell, devoted to hearing. So here we see the blue membranous labyrinth inside of the bony labyrinth. There's perilymph fluid in the bony labyrinth, so surrounding the membranous labyrinth, but inside of the membranous labyrinth, the fluid is endolymph. Then when we break down that membranous labyrinth, it's divided into the vestibular organ for equilibrium and the cochlear duct for hearing. The vestibular organ for equilibrium is divided into the vestibule and the semicircular canals. The semicircular canals are detecting rotation. The vestibule consists of two sacs called the utricle and the saccule. And these structures detect the effect of gravity on us, okay? Whether we're going up or down, and linear acceleration. All right, well, you do a cross section through. We see the bony labyrinth. Then inside of there is the perilymph. It's fluid. And then we have the membranous labyrinth. Inside of that is endolymph. So to the vestibular organ, having to do with equilibrium, detecting rotation, detecting if we're going up or down, detecting if we're going forward or backwards. It consists of the vestibule and the semicircular canals. The vestibule has two sacs inside of it, the utricle and the saccule. So let's talk about equilibrium. Detected by the vestibule. Again, detecting static equilibrium, just us being still, but staying upright, okay, not toppling over. Detecting movement, either up and down, or forward and backward. Detecting the orientation of the head, tilting, side to side, forward and back. Inside of the vestibule, we have these two sacs, the saccule and the utricle. The saccule is going to be continuous with the part of this structure, the membranous labyrinth that's devoted to hearing, the cochlear duct. The utricle is continuous with the semicircular canals, part of the vestibule that detects the, part of the vestibular organ that detects rotation. The vestibular organ is the vestibule and the semicircular canals. The saccule and utricle are the sacs, the membranous sacs inside of the vestibule. Okay, so here we are at the vestibule. And again, draw a line through here. This side is devoted to equilibrium. This is the vestibular organ that consists of the vestibule and the semicircular canals. This side is devoted to hearing. This is the cochlea. Okay. The vestibule consists of these two membranous sacs. The utricle, that's continuous with the semicircular canals, and the saccule that's continuous with the cochlea. Both the saccule and utricle are going to house the receptors for equilibrium. These receptor regions are called the macula. We have a macula in the saccule and a macula in the utricle. The macula is the receptor region detecting the position of the head, helping to control posture, responding to gravity, going up and down, or accelerating, or linear. The macula and the utricle, remember the vestibule, consists of the saccule and the utricle. In both the saccule and the utricle, we have macula, receptor regions for equilibrium. The macula and the utricle respond to horizontal movements. So side to side, back and forth, tilting of the head, okay? The saccule, the macula in the saccule is detecting vertical movement, up and down.
Yeah.
See how it does that? The macula, one in the saccule, one in the utricle. Two sacs, side of the vestibule. Contains sensory neurons called hair cells. And the sensory receptors, these special hair-like structures, stereocilia and kinocilia. The hair cells will get an electrical impulse that then gets transferred to the vestibulocochlear nerve, cranial nerve 8. The stereocilia and the kinocilia are the sensory receptors for equilibrium. They extend from the tops of the hair cells. And then they are going to be embedded in this membrane on top of them. This membrane is called the otolith membrane. It has it's a gelatinous matrix that has all these stones in it. These calcium carbonate stones. Let's see what that looks like. Here we are. These are the hair cells. Okay. The receptor cells. And these are the sensory receptors. Okay. The kinocilia and the stereocilia. Okay, so we're going to see those. When there is movement of the head, here is that otolith membrane, this gelatinous membrane with these calcium carbonate stones embedded in all this in our inner ear right now. We're sitting here. As we move, this membrane slides. Okay? We tilt our head, we slide. Everybody just slide your otolith membrane. And that bends these hairs. And that's going to cause an electrical impulse to be generated in the hair cell that will be transferred to these branches of cranial nerve 8. Send that information. The head is moving to the brain. Look deeper into these things. So here is the macula, histologic, the hair cells, the sensory receptors, or the hair cells, and then the sensory receptors, those kinocilia and the stereocilia, we'll see those, are embedded in this otolith membrane that sits on top of those hair cells. Gelatinous membrane with these tiny calcium carbonate stones embedded in it. We call them the otolith crystals. otolith crystals, the electron microscopy, that gelatinous matrix that they are embedded in, the macula. Here are the hair cells on top, or excuse me, the stereocilia and the kinocilia sitting on, coming out of the top surfaces of hair cells. So we have stereocilia, a whole bunch of stereocilia, plural, with always one kinocilium. And these aren't cilia that wave like other cilia we've learned about on our tissues, right? So they're not waving, they're actually still. They get bent. Whenever that macula, that membrane moves, slides, they get bent. And that triggers an electrical impulse. Stereocilia with a kinocilium. When we move up or down, the otoliths that are resting on top, right? So when we move up, the otoliths press down on our stereocilia and kinocilia. When we move down, that otolith membrane lifts up off of them. Yeah, get that? Yeah? Okay. Thank you, Dad. Appreciate that. All right, and then that signals to the brain. Oh, we're moving up. Oh, we're moving down, like in an elevator. When we tilt our heads, that membrane is sliding over those stereocilia and kinocilia. That tells the brain, okay, our head is moving side to side, or forward and backwards. We're moving forward and backwards, tilting our head forward and back. That otolith slides forward or back or forward, right? If we're neutral, and gravity is pressing down on that, right, and the brain can tell, right, that we're at some static equilibrium. We're just being nice and neutral and still. But if we move down, it's gonna lift up off. We move up, gravity is gonna press down. Or tilt, slide back and forth. When that otolith membrane either presses down on these receptors, sensory receptors, the stereocilia and the kinocilium, when those get disturbed by the movement of the otolith membrane, they generate an electrical impulse. And then those hair cells, right, from which the kinocilium and stereocilium are coming off the top of, right, they're attached to. I'm gonna release neurotransmitter. Okay, I'm gonna release a neurotransmitter. If we bent the stereocilia, and if we bend the stereocilia, depending on which direction they bend, and towards the kinocilium that's there, or in the opposite direction of the kinocilium, different things will happen. We'll depolarize. That just means we're increasing the amount of neurotransmitter we're secreting. Or we'll hypopolarize. That means we're going to reduce the signal. Okay? You'll learn more about that when you get to physiology. But this, either secreting more neurotransmitter or secreting less, is telling the brain two different things. The different ways our head are moving. The different changes in the position of our heads. And when we have the release of neurotransmitter, okay, we're going to fire down cranial nerve 8, the vestibular division of the vestibulocochlear nerve. The cochlear division is devoted to hearing. The vestibular branch is devoted to all of this, detecting what's going on, and then the brain will adjust our equilibrium accordingly, depending on what it is being told is happening with our heads or with our bodies. Are we moving forward? What are we doing? Are we going to start spinning? We're going to have some other signaling going on. everybody, okay? Spinning is detected at the semicircular canals. This is some dynamic equilibrium. That means we are rotating, it's a moving equilibrium. The semicircular canals consist of these three canals, superior, inferior, and horizontal one. And you'll see on the, to be able to determine who's that, which ones those are. We're just considering the semicircular canals all together. The semicircular canals are bony labyrinth, still some bony labyrinth, the membranous labyrinth inside. And the membranous labyrinth is making these membranous ducts that travel the contours of the bony labyrinth. Remember, the utricle of the vestibule is continuous with the semicircular canal, so we get some communication. At the beginning of each one of these semicircular canals, these ducts, there's this bulge, this swollen bulge that we see right there at the utricle, attached to the utricle of the vestibule. Those bulges are called ampulla. So here they are, semicircular canals. They have these bulges at their ends that are continuous with the utricle. And then the membranous labyrinth inside is this duct traveling inside that bony labyrinth. Inside of the ampullas, now we're gonna find the receptor region for this type of equilibrium. This is called the crista ampullaris. You have one in each one of those ampullas, those bulges at the ends of the semicircular canals, anchored to the utricle. It's the crista ampullaris that's going to be firing electrical impulses due to changes in velocity and spinning. That's what's inside of an ampulla. That bulge at the end of a semicircular canal. Now we're inside. It's the membranous labyrinth inside, so there's endolymph, fluid in here. Here is the crista ampullaris structure here. Sitting on top of that is a structure called the cupula. You can see all that via electron microscopy. Looking inside the ampulla, we can see the cupula that sits on top of the crista ampullaris. There. Okay, let's look at crista ampullaris a different way. Histologically, there is the crista ampullaris structure here, and there's the cupula sitting on top of it. And we're inside of that bulge, the ampulla, at the end of the semicircular canal. There it is again. Here is an ampulla of a semicircular canal that is continuous with the utricle of the vestibule. There's a lot of terms that you know, we're gonna get it down. Practice, practice, and what? What are we doing after that? Practice is more. Here we go, inside of that ampulla. Here is the crista ampullaris. And the cupula sitting on top of it. We are inside of the ampulla at the end of the semicircular canal. It is filled with endolymph, continuous with the utricle. Oh, what do we find in the utricle? There's the macula. We have that otolith membrane resting on top of the stereocilia and kinocilium of the hair cells. Here we're detecting a different type of equilibrium. Here we're going to be detecting a different type of equilibrium. Okay? All devoted to detecting equilibrium. This is the membranous membrane that's surrounded by perilymph. The membranous labyrinth surrounded by perilymph. So here's the bony labyrinth. Remember what bone looks like? Does that look familiar? No? All right. The crista ampullaris. Contains the sensory neurons, more hair cells, and the sensory receptors, more stereocilia and kinocilia. There's nothing different there. And the hair cells are doing the same thing here. But here, the stereocilia and the kinocilium aren't embedded in macula, that gelatinous membrane with stones in it. Here, the stereocilia and the kinocilium are embedded in the cupula, that's sitting on top. Still some gelatinous stuff. But when those stereocilia and kinocilia get moved, they're gonna trigger an electrical impulse that fires from the hair cells and gets delivered in the synapse at the fibers of the vestibular cochlear nerve, particular the vestibular branch. When we are rotating, that endolymph inside of the membranous labyrinth of the semicircular canal spins. Okay? It starts to move. And when it moves, it causes the cupula to move and bend those stereocilia and kinocilia. When we rotate to the right, those sensory hairs first bend to the left because of inertia. Inertia is the force against movement. Okay? So first, as you're going to the right, those hairs are going to bend to the left because of inertia, the resistance against movement. Okay? And it's because of the endolymph. The endolymph is doing this. Okay? It's going to first bend the other way of your movement. But then, as you continue to spin, eventually that endolymph is going to start to spin with you, catch up with your spin. Okay? As you start, it's going to lag behind, go the other way. But eventually, it's going to start moving with you. Okay? Your spin is going to catch up with your movement. The movement of the endolymph is going to catch up with what you're doing. When it catches up, the cupula is no longer moving. Okay? Because that endolymph is moving with you. So the cupula doesn't get moved in any way. It's only when you first start spinning, and that endolymph goes the other way, that cupula gets moved. Until finally, everything catches up with your movement and the cupula will no longer be affected. And that stops the sensation of rotation. When you move in a circle like that, and then you stop abruptly, the endolymph in that membranous labyrinth doesn't stop moving. It continues to spin. And that's what causes us to be dizzy. When we stop moving after spinning, that endolymph in our semicircular canals is still spinning. Until it stops, we're going to feel dizzy. Okay.
Can that be trained
Well,
for like figure skaters?
yeah, look at that. Figure skaters can adapt to all of that. That's why they can spin like crazy. Okay. So when you're at rest, that cupula, right on top of the hair cells. Okay? When you start to spin, the cupula, if you're spinning this way, the cupula first gets bent in the other direction because there's fluid in here. Okay? And it first moves in the other direction. Okay? And then that is going to fire a signal that we're rotating. But then, eventually, that the cupula will start to, the endolymph will start to move with you. Okay, so it'll start to spin with you. But then, as you slow down, that endolymph is still moving, and so the cupula still is getting bent again. And when you stop abruptly, it's still moving and you feel dizzy. But figure skaters can adapt to this. Doing it enough, they kind of it doesn't make them dizzy like it would us. We did it for the first time, spit that fast.
So with their training, it allows them to get used to it.
Yes. Okay, so with this, we have activation of that crista ampularis. The cupula here's the crista ampularis. Cupula is sitting on top of the crista ampularis. That's where we have hair cells with their stereocilia and kinocilium. And it's going to be responding as that cupula is bending. Those hair cells get bent. And we'll get, again, a firing of neurotransmitter, the rapid firing of impulses, or not so rapid. And that will tell the brain what we're doing as far as spinning. When those hair cells are bent in the opposite direction of our spin, we get fewer impulses. But all of this information about spinning and whether we have stopped spinning, the fluid is still moving, so we feel dizzy, all of that, the neurotransmitter that's getting secreted, and will then go and bind at the dendrites of the vestibular branch of cranial nerve 8, whose fibers are right there at the base of the hair cells. That's how the vestibular branch is going to get the information from the sensory receptors for this spinning, that we are spinning, or that we have stopped spinning. Everybody okay? Some folks suffer from vestibular nystagmus. And we all can actually suffer from this. So vestibular nystagmus is when we are spinning. You can actually see this in people. When you spin and then you stop, our eyes actually are moving side to side. So the eyes will drift opposite to the way that you're rotating. And this is because the eyes are trying to figure out which way are we going. We're spinning, now we're stopping, and they have to try to catch up with what our head is doing. And so you'll get this movement in our eyes called vestibular nystagmus. I actually had a cat who had this.
Who usually
For
gets
eyes.
that? Who usually get that?
Oh, it's,
I feel like when you
Is
see
it
little kids
hereditary?
spin, you can see their eyes do that.
yeah. Oh, no, this is gonna, it could be anybody that gets this. You said military? Hereditary, yes, it's hereditary. So anybody, this can happen to anybody. But this here, this can happen to all of us. All we gotta do is spin. And stop and somebody's eyes will be moving the opposite of the way that they were spinning. Some people hereditarily have a neural dysfunction where it's always, have you seen this actor? Have
Yes.
you seen him? Well, you look at his eyes when he's on those screens, they move.
And continuously move?
You know the movie Constantine with, Keanu Reeves?
Yeah.
He's in that. Watch that.
I don't
You'll
have
see
to
a
watch
lot
it.
of it.
I see it,
I like that.
pay attention to that.
But yeah, that's the only movie I can think of. That he's
I gotta
in.
look it up.
But your nephews.
That was him. Trying to do it, no, that's him. So he has just a neurological issue where it's always happening in him.
Oh, wow.
We can make it happen to us just by spinning.
Bro. This? Too big? How much to put on there, bro?
I don't
It's
think
for
that,
life.
I mean, he's acting,
I ain't going.
he don't look like he's dizzy. But I don't think that you actually, he
That's-
feels dizzy.
This is
It's just
all
his eyes.
lab
Maybe they
13.
would've-
I'm already done.
I mean, I don't know.
I have two more labs.
Eyes, I mean, I don't know how, I don't know how dysfunctional his life is because of it. Okay? Or I don't know how anyone's life is dysfunctional because of that. That's not something that I read about. But definitely feel free to see, if someone suffers from vestibular nystagmus, can they drive? That's a good question. Okay.
Send them
Motion
a group
sickness
chat. Like,
is
if you
when
take
we have these
screenshots,
sensory
important ones.
mismatches. Okay? So our visual input is differing from our equilibrium input. So one, example is if you're in a car, but you're looking down like reading or something, right? You're not looking at the road, okay, you're just looking down. So your body is moving, and that's what your brain is sensing. But your eyes are having this different visual cue, and that causes motion sickness. So you all you gotta do is look up and look out, and let your eyes do what your body is doing. Kind of looking forward and accelerating forward too.
I get motion sickness in the car if I don't drive, if I'm a passenger.
Mm. Okay, so we have this whole pathway in the brain. Once that the vestibular branch of the cochlear nerve gets its signal, okay, that's gonna send that information to the brain stem. Okay, and at the brain stem, we can get some reflexes in response to imbalance. Okay, so if our brain is, or if the signal tells right there at the brain stem that we're tipping over, maybe the reflex will be to catch yourself. It's a muscle movement, catch yourself, right? But then we're going to, this electrical impulse will be delivered to different sets of neurons, okay? Go from the brain stem, the cerebellum, we'll go to different sets of neurons, and finally, we will be in neurons that are in the vestibular cortex. The primary vestibular cortex in that post-central gyrus, remember him? Where we are talking about somatosensory stuff, sensation at the body, right? So we get this whole transfer of these electrical impulses from cranial nerve 8. That's the first order of neurons, okay? To a whole new the nuclei of neurons, the vestibular nucleus. And that's the next step in the traveling of this electrical information via neurotransmitter, right? Transmitter gets released from these guys onto these guys. And then they will innervate or release neurotransmitter at a third order, another set of neurons in the thalamus that will then deliver that information, all right? Thalamus relay sensory information. All up in the primary, the vestibular cortex is in the postcentral gyrus. Okay, ready to do hearing? Here we go, let's go check out the cochlea. The spiraling, conical, bony chamber with membranous labyrinth or bony labyrinth with membranous labyrinth inside of it, very tiny, inside of our ear, embedded in that petrous portion of the temporal bone. Okay. It's going to extend from the vestibule and those semicircular canals, extends from the vestibule, and as it extends from the vestibule, it spirals. And at the very center of the spiral is its apex. The cochlea is coiling around this bony pillar. Is at the center of it, called the modiolus. It stops at its apex. Here we're gonna find sensory neurons that are associated now with the cochlear branch of the vestibular cochlear nerve. In here, in the bony labyrinth here, you have the membranous labyrinth inside of it. That membranous labyrinth here is called the cochlear duct. And inside the cochlear duct is the organ of Corti, also called the spinal organ. And this is the structure inside the cochlea where we are getting our sensation for hearing, where there's going to be hair cells, sensory receptors for hearing. So let's make our trip into the cochlea. Okay, here we are now. So we already were here, that's the vestibular organ with the vestibule, one type of equilibrium, up, down, forward, backwards, head tilting side to side, or our semicircular canal spinning. Now we're making our way into the cochlea. That winds all around until it gets to its tippy top apex. And it winds around the modiolus, this bony pillar. Okay, so we'll see this in our model. The cochlea. A beautiful, someone wanted a beautiful electron micrograph of the cochlea. Inside of the bony labyrinth of the cochlea is the membranous labyrinth, the cochlear duct. Inside of that is the structure for sensing hearing. Okay. The organ of Corti. Here is the inner ear. And we can see the organ of Corti. This person labeled it fluorescently and imaged it. My alma mater, you do. All right, let's look at the cochlea. We have some chambers here. We have some questions about these chambers in the lab. Here they are. The cochlea is divided into 3 chambers. So that membranous labyrinth is divided into 3 chambers. The scala vestibuli, the scala media, and the scala tympani. The scala vestibuli is going to start at that oval window, the entrance into the inner ear. That small bone number 3 in line, stapes, is resting up against that oval window. Inside of scala vestibuli, this chamber is perilymph. Then we have scala media. Okay, so I'm going to take that back. All of this is not membranous labyrinth. This is perilymph is surrounded by bone, scala vestibular, the bony labyrinth. Then we have the scala media, also called the cochlear duct. That is membranous labyrinth. Inside of that is endolymph. This chamber, scala media, lies between scala vestibuli and scala tympani. Scala tympani contains perilymph, so that's surrounded again by bone. And it's going to end at what's called the round window. That's the exit from the cochlea. Scala tympani and vestibuli are going to be continuous with each other. Okay. When we get to the apex, it's also called the helical trema. You go into one and into the other, one and into the other. All right. Let's understand where we're at. Looking inside of the cochlea. Now look at these chambers. And we have the membranous labyrinth running through the bony labyrinth. The membranous labyrinth here gets a name. The cochlear duct, also called scala media. Endolymph is in there. And then we have our other chambers, scala vestibuli and scala tympani. Here's that modiolus, this bony structure here, around which the cochlea is winding. As it winds, let's go this way, we get to the helical trema, that apex. And this is where the scala vestibuli and tympani are continuous with each other. We go into one and the other. Okay. Get this is from the model. Ours is not color coded, unfortunately. But these structures are on our lab practical list. So we do the lab, we'll talk about how we'll see them. Our model doesn't make it nice and color coded like this, so we'll use pictures. All right, I'm in the lab. From the lab. Here, or this lecture. Here we go. All right, there's that. Let's go into, let's go. Okay, so here we are. There is the membranous labyrinth, also called scala media. That's the chamber's name. Also called the cochlear duct. Endolymph inside there. Then we have scala tympani and scala vestibuli. Perilymph inside of these.
How can you tell the difference when you're, I mean, if it's not color coded?
Just by their positions, so you can kind of compare our, and that's why we're not gonna use the model, because it's not color, our model. We'll use the pictures, where we have some color helping us, and we don't have a key, and I don't even know if it's numbered, a letter like this. Yes, it might be.
The model doesn't have any numbers. I'm not going to fix that.
Right. So there are the chambers. Here's scala media, vestibuli and tympani. And media. Okay, let's go into the cochlear duct now. Inside of scala media, because that's where we're going to find the vestibular membrane. The vestibular membrane separates scala media from scala vestibuli. The vestibular membrane separates scala media from scala vestibuli. Okay. The floor of the cochlear duct is called the basilar membrane. This is where we have the organ of Cordy sitting on top of the basilar membrane. And I can take that back. Here's the vestibular membrane. Let's label it. This is the basilar membrane. Sitting on top of that, we're gonna see this up, blown up a little bit better. Sitting on top of that is the organ of Cordy. All about hearing. This is where we're gonna get branches from cranial, the cochlear branch now, of cranial nerve 8, the vestibular cochlear branch, extend from the organ of Cordy. So whatever happens in the organ of Cordy is gonna disturb or activate the cranial nerve 8, the cochlear branch of cranial nerve 8, and send to the brain and what we're hearing, information about what we're hearing. All right, let's look inside one of these. There it is, scala vestibuli, scala tympani. Here is the vestibular membrane that separates scala media from scala vestibuli. Here is the basilar membrane. It's the floor of scala media, also separating it from scala tympani. Sitting on top of the basilar membrane is the organ of Cordy. We're gonna have hair cells, stereocilia and kinocilia, doing their thing when stuff happens. And we have branches coming from the vestibular cochlear branch of the vestibular cochlear nerve. Is gonna be right here, ready to get some neurotransmitter from those hair cells. Wait. Okay. Let's make our way around. There is scala vestibuli, scala media, scala tympani. A ganglion. Okay, so that is a group of neuronal cell bodies in the peripheral nervous system for cranial nerve 8. So have fibers that are reaching over to the organ of Cordy. Let's get even closer. Okay, scala vestibuli, scala tympani, scala media. This is where we want to be. Let's see the organ of Cordy, hair cells and so forth. You guys remember this? We were looking at simple squamous epithelium. This is where this comes from. But this is the vestibular membrane. So it's not a little thin strip. It's an entire membrane. You just got to slice through it. So we're looking at the edge of that membrane. Okay? It is a membrane. So it's a structure. We're just looking at the edge of it. Okay? Let's do it again. Scala vestibuli, scala tympani. Let's knock those out. Now let's go to where the fun stuff happens. Scala media. The ceiling of scala media is that vestibular membrane. The floor of it is the basilar membrane. Sitting on top of the basilar membrane is the organ of Cordy. Where we have the ganglion from cranial nerve 8, where the cell bodies of neurons are, and it's fibers. They're axons are coming to be rest up on these hair cells and wait for some sprinkling of some neurotransmitter. Once again, okay, practice, and practice some more. Scala vestibuli. Perilymph in here. Scala tympani. Perilymph in here. Scala media. Endolymph in here. The vestibular membrane. The basilar membrane. sitting on top of that is the organ of Corti. It has a little membrane itself called the tectorial membrane. Let me add something else. So, the organ of Corti. The organ of Corti. Also called the spiral organ. It's sitting on top of that basilar membrane. Sitting on top of it between scala media and scala tympani. That basilar membrane between those two chambers. Organ of Corti sitting on top of basilar membrane. It has hair cells. Those hair cells have lying over them the tectoral membrane. This here. When you dissect it, it kind of snaps away, but it's stretched out really over these hair cells, that tectoral membrane. When sound waves come in, they cause a distortion at the tectoral membrane, and that stimulates the organ of Corti. That stimulates those hair cells. When that tectoral membrane on top of it, always a membrane sitting on top of these hair cells, gets disturbed in some way. Either it's the otolith membrane sliding across them, right? Or it's the ampulla bending them in the semicircular canals, or the cupula, bending them in the semicircular canals. Here is the tectoral membrane. And when it gets disturbed by waves, it's gonna stimulate those hair cells. We're gonna fire some information via electrical impulses. Starting with release of neurotransmitter. Okay, let's make our way around here. There's that basilar membrane, the organ of Corti sitting on top of it, where we have hair cells that have their stereocilia. Here's that tectoral membrane that's lying on top of the hair cells. Remember, there's endolymph in here. So when waves come in, or when waves start, make their way here, it disturbs this membrane. That moves those hairs. That fires a signal in these hair cells. And then they secrete neurotransmitter at these fibers coming from the cochlear branch of the vestibular cochlear nerve. That is sending information about what we've heard to the brain. Okay. Organ of Corti. Tectoral membrane. Again, this is a slice through all this, but this is a membrane, so it's not that little piece of something, it's a membrane. We just see a slice. So we're looking at the edge of that membrane. When we dissect this, that tectoral membrane kind of snaps back. It's not stretched out over the hair cells here of the organ of Corti. Here's his hair cells. Tectoral membrane would be lying here, kind of in contact with the stereocilia that you can make out. Okay, we're gonna get even closer. Here we go. Here, tectoral membrane is flipped all the way back. And it should be lying on top of these hair cells of the organ of Corti. Okay, here is a little bit in better position. Tectoral membrane lying on top of the hair cells of the organ of Corti that has its stereocilia. That tectoral membrane is in contact with those stereocilia. All right, what happens when sound does its thing? What is sound? Sound is a wave, a pressure wave. It comes in, it travels this way through solid things like the wall, through liquid, through gas. Sound wave can travel this wave of pressure. That pressure can cause vibration in objects. sound wave can move in all directions. And it's drawn as a sine curve, right? So up and down, up and down. So sound waves look like this, a sine curve, where the frequency and wavelength mean things. The frequency is the number of waves that pass a particular point at a given time. So this will be high frequency sound, low frequency sound. Pitch is how we perceive those frequencies, high or low. Wavelength is the distance between two crests. The shorter the wavelength, the distance between two crests, the higher the frequency. The amplitude of a wave is perceived as loudness. Higher the amplitude, greater the amplitude, louder the sound. Sound is coming in to our auricle like a wave, as a pressure, wave of pressure coming into the auricle of your ear. All this curviness, all this stuff in our ears is capturing those sound waves coming in, directing them into our external acoustic meatus. We get in there, still traveling like a wave, and then we hit the tympanic membrane. Now that sound wave causes the tympanic membrane to vibrate. Behind the tympanic membrane, the ossicles start to vibrate. Malleus first, then incus, and then stapes, right at the oval window, the entrance into the cochlea. Inside there, we have that fluid. Now those waves, sound waves, that have been changed into vibrations of the tympanic membrane that have traveled through the ossicles. Now we're gonna get waves in the fluid because of that vibration in the fluid inside of the cochlear duct, or inside of the cochlea itself. Those pressure waves now are going to move through the perilymph that's in scala vestibuli. We go through, so the oval window takes us into scala vestibuli. And now we're gonna have waves through the perilymph of scala vestibuli. This is what's happening as I'm talking to you. Happening inside of your scala vestibuli right now. I'm making waves in your perilymph. You're welcome. Then that sound waves, if it's out of our hearing range, because we can't hear every frequency, right? So waves at frequencies below our threshold of hearing will travel around that cochlea in the perilymph of vestibula, scala vestibuli, all the way to the tip, the apex of it. Okay? And then turn and travel through scala vestibuli, or tympani. And then make exit out the round window. This means that it never gets to the organ of Corti. It comes in, scala vestibuli, turns and goes out of scala tympani. It comes in the oval window, these waves, and then goes out the round window, never getting into the cochlear duct to disturb that temporal membrane and the hair cells, and that's for sound we can't hear.
Professor, do you know why children have like they're more sensitive, I guess, to the wave? Because I know there's some sound waves that adults can't hear, but children can.
I think that's just by age. As we age, we get a decrease in hearing age, right? So our sense of hearing ages.
All the damage we
Yeah, or if you get damage, and had those earphones on in the 80s too loud.
Yeah.
Walkman. Those of us who remember Walkman.
think it's worse now than,
Do you? Okay, so there are some sound waves coming into the external acoustic meatus. Hits the tympanic membrane, causes vibrations. Those vibrations travel through the ossicles. And right at the opening to the cochlear, at the oval window where stapes is right up against it. We get those waves now transferred into the liquid, the perilymph of scala vestibular. And they travel, they travel, they travel, they travel. This is for a frequency that we can't hear. They travel to the apex. And here, this is where scala vestibuli is continuous with scala tympani. Now they're in scala tympani. They travel, and they travel, and they travel, and they travel out of the round window. Back into the inner ear, where they've lost their strength so much that they're unable to cause any vibrations in the tympanic membrane. They just dissipate. Okay? That's for stuff that you can't hear. All that is, how do the, explain the pathway of sound wave? Here you go. All spelled out for you. Practice, practice, and practice some more. Now, for stuff, let's hurry up. For stuff that we can hear in range. Now we're gonna disturb scala medium. We're gonna disturb the basilar membrane. We're gonna disturb the tectoral membrane. We're gonna get into scala media now. Or we're gonna cause some disturbance inside of scala media. Okay? The sound waves are going to go through the cochlear duct for frequencies we can hear. They're gonna vibrate basilar membrane where the organ of Corti is sitting. Now we're moving the organ of Corti. Uh-oh, that's going to cause those hair cells and their stereocilia to be displaced from the tectoral membrane, right? Sitting, that is on top of them. That is going to cause stimulation of the cochlear branch of cranial nerve 8. They send those impulses without hearing to the brain. Okay, so what we can hear comes in. Okay, still comes into the scala vestibular. But with the frequencies we can hear, we disturb the scala media and the organ of Corti inside of there. And when we do that,
Could you change it back?
right there. When we do that, we'll disturb the organ of Corti that's inside of here. So these sound waves that are in frequency that we can hear go into the cochlear duct, into scala media, and vibrate the basilar membrane in there, and cause the displacement of the hair cells and their stereocilia on top of them, away from the tectoral membrane resting on top of it. Okay? Okay, so high frequency sound will displace the basal, the basilar membrane kind of near the oval window. Medium frequency sounds will travel a little bit and then displace the cochlear duct and therefore the basilar membrane kind of farther down. So this is the cochlear duct but stretched out instead of spiral. Okay, lower frequency sounds will displace further down the cochlear duct. Sounds we can't hear won't even displace it. They'll just come on out the round window. Okay.